what if everything you've been taught about aging, like that it's just natural, inevitable, just a fact of life, what if all of that is actually completely wrong?
Yeah, I mean, it's a massive question. It really challenges the absolute baseline assumptions we make about biology and, well, our whole lives.
Exactly. And that's what we're testing today. Welcome to this custom-tailored deep dive. We're looking into some totally paradigm-shifting research from Harvard genetics professor Dr. David Sinclair, specifically deconstructing his information theory of aging.
Right. And the goal here is to really shift from viewing aging as this, like, passive, inevitable wear and tear thing to treating it as an active underlying disease, like
Yeah.
the actual root cause of things like Alzheimer's or cancer.
We're going to get into the cellular mechanics, the wild lab tech that's literally reversing the age of primate tissue, and the daily habits you can start today to potentially stay vibrant well into the 22nd century.
It's super exciting stuff.
It really is.
Yeah.
But to really grasp the gravity of this whole pursuit, we kind of have to look at where it started. And it started with a really emotional story from when Sinclair was, what, 4 years old?
Yeah, 4 years old. His grandmother, Vera, who was a survivor of World War II, she essentially sat him down one day.
Right, because he asked if she'd always be there to protect him, right?
Exactly. And she gave him this incredibly harsh truth for a toddler. She just flat out said, no, everything dies. She told him his parents would die, his cat would die, and, eventually he would die too.
Which is just, wow, that just shattered his entire worldview.
Right. I mean, usually that kind of existential dread in a 4-year-old just sort of fades into the background as we grow up. We just accept it.
Yeah, we totally just accept it.
But for him, it didn't. It evolved into this 30-year career at Harvard. Like, if aging is this universal process, killing over 100,000 people every day, accepting it just because we've labeled it natural, that just became scientifically unacceptable to him.
Okay, so to understand how to actually reverse aging, we first have to figure out what aging actually is. Like, we need to completely throw out that old mechanical wear and tear model. Like, your body isn't just an old shoe wearing out.
Right, exactly. And the critical focal point here is the epigenome.
The epigenome. Okay, break that down for us.
So we used to think DNA degraded over time, but we know now that DNA is just the static hardware, right? The genetic code itself doesn't actually degrade the way we thought.
Okay, so the hardware is fine.
Exactly. The real focus is on the software running the show, which is the epigenome. Specifically, these things called methylation patterns. These are the chemical markers that tell a cell, hey, you're a nerve cell, or you're a skin cell, or, a liver cell.
Okay, so they give the cell its identity.
Yep. And managing that entire software system, you have these things called sirtuins.
Ah, the sirtuins. The longevity genes.
Right. They act like molecular conductors, but
Yeah.
they actually have a dual mandate, two jobs, which is where the whole biological vulnerability comes in.
Okay, so what are the two jobs?
Job one is silencing genes to maintain that cellular identity we just talked about. But job two, they are the primary responders to broken DNA, to double-strand breaks.
Broken DNA. Like, from what?
Well, just everyday environmental stress, cosmic radiation from, like, a transatlantic flight, heavy metals, ultra-processed food, or honestly, even just standing next to a giant loudspeaker at a rock concert.
Wait, really? A loud concert can break my DNA?
Yeah, it literally causes microfractures in your chromosomes.
Wow. Okay, so with chromosome fractures, what do the sirtuins do?
They drop everything. They basically abandon their post managing the cell's identity, and they rush over to repair the structural damage. It's biological triage.
They prioritize the emergency. Makes sense.
Right, they fix it. But here is the massive flaw in the system. When they finish the repair and make their return journey, a small percentage of those sirtuins, they get lost. They don't make it back to their original epigenetic spot.
Oh, I see.
Yeah. So over decades, after millions of these little micro-emergencies, the patterns become chaotic, the software degrades.
And that's what causes the identity crisis of the cell.
Exactly. A nerve cell basically forgets it's a nerve cell and starts acting a little bit like a skin cell. And that loss of function is why our hair goes gray, why our skin wrinkles, why organs start to fail.
That makes the vinyl record analogy from the research make total sense.
Yeah.
It perfectly visualizes this information loss.
Yeah, it's a great way to look at it.
So, like, the DNA is the physical groove pressed into the vinyl, right? It contains this beautiful symphony. And the music doesn't actually disappear. Aging is just the accumulation of physical scratches on the surface of the vinyl that makes the needle jump around.
Right.
But the symphony, the actual music, is still completely intact underneath all that noise. So, I mean, the obvious question you have to ask is, can we just polish the scratches out of the vinyl?
And the incredible answer is yes. A backup copy of the original epigenetic pattern, like that youthful software, it still exists in every single old cell. It's just sitting there waiting as an accessible restoration point.
Okay, so if the original software is still archived in the cell, how are scientists actually accessing it? Because Sinclair's lab in Boston is doing this right now, right? With a virus.
Yeah, they use a viral delivery system called AAV2. It stands for Adeno-Associated Virus 2.
Delivering genes with a virus just sounds kind of scary.
Well, viruses have evolved over millions of years to be basically the perfect delivery vehicles. They're incredibly efficient at getting inside cells. So virologists actually hollow out the virus. They strip away all the disease-causing parts.
So they just use the shell, like an empty biological FedEx truck.
Exactly, a microscopic FedEx truck. And inside this empty shell, they load 3 very specific genes, OCK2, SOX2, and KLF4.
And those are 3 of the Yamanaka factors, right?
Yeah.
The ones famous for turning adult cells back into stem cells.
Yep. But before they started shooting these rejuvenating genes into things, they had to prove they actually understood how the scratches on the record worked in the first place.
Right, they had to prove the information theory. And they did that with the ICE mice.
Yes. The IC mice, it stands for inducible changes to the epigenome. Basically, they introduced a gene from a slime mold.
Wait, slime mold?
Yeah, Physarum polycephalum. It's brilliant, actually. This specific slime mold enzyme makes very precise cuts in the mouse's DNA, but it explicitly avoids cutting the actual coding regions, so it doesn't mutate the genome or cause cancer.
Oh, so it just perfectly simulates the everyday stress we were talking about.
Exactly. It purely simulates those double-strand breaks caused by modern life. So they artificially accelerated that triage response. They forced the mouse's sirtuins into a state of chronic panic.
So what happened to the mice?
Well, for 3 weeks, they induced these breaks, and outwardly, nothing happened. But 10 months later, those mice looked incredibly old compared to their twin siblings. Graying fur, hunched backs, failing organs.
Wow, so the distraction of the sirtuins explicitly drove the aging process. They proved they could scratch the record.
Right, and once they proved that, they deployed the AAV2 FedEx truck with those 3 Yamanaka factors to polish the record, and they targeted primates that had gone completely blind.
Because the optic nerve was damaged due to aging. Why start with the eye, though?
The optic nerve is brilliant because it's part of the central nervous system, so it's brain tissue, but it's physically isolated in a little enclosed space. It's much safer to observe.
Makes sense.
Yeah.
So they inject the eye. Does the virus just do its thing immediately?
No, delivery was just step 1. They engineered those 3 genes to need a switch. They respond to an antibiotic called doxycycline. So you give the primate the virus, and it just sits there, totally dormant.
Until they give them the antibiotic.
Exactly. The doxycycline acts as a molecular trigger. It turns the rejuvenation genes on.
And by turning those genes on, the primate's optic nerve cells literally reset their biological age by like 75%, right?
Yeah, about 75%. The cells regenerated, they reconnected to the brain, and they effectively cured the blindness in these monkeys.
It's wild that they're submitting this to the FDA for human trials this year.
Mm-hmm.
But I mean, logically, pushing a cell's age backwards feels incredibly risky. Like, wait, if you reset a cell, how does it know where the breaks are?
Right, why doesn't it go too far?
Yeah, why doesn't it regress all the way back to an embryo or turn into a tumor?
That's where leaving out the 4th Yamanaka factor is crucial. There are normally 4. The one they omitted is called c-Myc.
Oh, okay, so c-Myc is the danger factor.
Basically. By leaving it out, the cell reaches a natural epigenetic barrier. The biological clock rewinds, but it hits a wall at that 75% mark. It gets youthful, but it remembers it's an optic nerve cell. It doesn't become embryonic.
That is just elegant. And it really forces us to rethink what chronic disease even is. Because if you rejuvenate the body and an old cell regains its youth, the whole environment that fosters disease just sort of collapses.
Oh, absolutely. We see this directly with oncology. Think about it. Cancer is arguably the ultimate cellular identity crisis.
Right, because it forgets it's part of a team and just starts aggressively copying itself.
Exactly. It's a zombie cell. But when Sinclair's lab applied this epigenetic reprogramming to cancer cells, the cells essentially woke up. The software reset, they remembered they were supposed to be healthy tissue, they realized they were mutated, and they just initiated apoptosis.
Apoptosis, that's programmed cell death, right?
Yep, they killed themselves. So reversing age doesn't just treat the symptoms of age-related diseases. It actually eliminates the biological foundation that those diseases require to exist in the first place.
Okay, this is all mind-blowing.
Yeah.
But I also know we are still years away from just picking up an AAV2 gene therapy prescription at the local pharmacy.
Hmm.
So what controls the scratches on our record right now?
Right, what can we do today?
Yeah, because the staggering consensus here is that 80 to 90% of our biological aging rate is dictated by lifestyle, not our hardwired DNA.
And the driving biological principle behind that lifestyle is called hormesis.
Hormesis, what exactly doesn't kill the cell makes it stronger?
Pretty much. It's the biological concept of adversity. It's the phenomenon where a low dose of a stressor, something that might kill you in a huge dose, actually provokes a beneficial, adaptive response in your cells.
And our modern problem is that we've completely trapped ourselves in abundance mode. I mean, we have 3 hypercaloric meals a day, air conditioning everywhere, we sit at desks all day. The cellular machinery just gets totally lazy.
Totally complacent. In abundance mode, your cell's energy sensors just tell the body, hey, focus on growth and reproduction. Times are good.
But we need to intentionally trigger adversity mode, right? To force the body to deploy its longevity defenses.
Exactly. When you introduce mild adversity, the body shuts down the growth pathways and diverts all that energy toward deep cellular repair. And fasting is the most accessible lever we have for this.
So skipping meals, like waiting until 3 p.m. to eat.
Yeah, narrowing your eating window. When you restrict caloric intake, you fundamentally alter your cellular chemistry. Specifically, the nutrient deprivation signals your body to synthesize more NAD.
Okay, NAD. Nicotinamide adenine dinucleotide. Let's connect that back. If the sirtuins are the conductors trying to fix the epigenome, NAD is the essential fuel they need to do their job, right?
Perfect analogy. Sirtuins literally cannot function without NAD. And the bad news is, our natural NAD reservoirs drop off a cliff as we age.
But fasting forces the body to make more of it.
Yes. And if you extend that fast, say to a 3-day water fast once a month, you push that hormetic response much, much further. You trigger something called chaperone-mediated autophagy.
Autophagy, that's like deep cleaning, right?
Exactly. The cell senses a severe nutrient crisis and panics in a good way. It begins hunting down misfolded old proteins and damaged mitochondria, and it breaks them down to recycle them for energy.
I always think of it like an overstaffed company during a massive economic boom. Like the employees are just lounging around, efficiency is terrible, mistakes are piling up.
They get comfortable.
Yeah. And then a recession hits. That adversity forces the executives to wake up, audit the company, fire the deadweight, and optimize everything just to survive. Yeah. So we basically have to trick our bodies into thinking a famine is happening just to keep our cellular maintenance crews awake.
That's exactly it. And evolutionary biology supports this perfectly. Think about animals that evolved without natural predators.
Like the bowhead whale or the bristlecone pine.
Yeah, or the Greenland shark. They developed extreme, staggering longevity. Because they weren't under the constant threat of a lion attack, their bodies could afford to invest heavily in long-term DNA repair mechanisms rather than just fast, frantic reproduction.
But humans basically removed all our natural predators.
We did. So we have to artificially synthesize that stress to activate those dormant repair systems.
And that artificial stress extends into how we move, too, because a casual walk or a light jog isn't enough to trigger hormesis, is it?
No, it's really not. The goal is hypoxia. You need to lose your breath. You have to push your cardiovascular system to the point where you are panting, where you literally can't maintain a conversation for several minutes at a time.
So that temporary lack of oxygen signals severe adversity to the body.
Exactly. And thermal stress operates on the exact same axis.
You mean like saunas and cold plunges?
Yeah. Exposing yourself to the extreme heat of a sauna or the acute shock of an ice bath, it forces the body to synthesize heat shock proteins. They act as molecular bodyguards, stabilizing other proteins and preventing them from misfolding under the stress.
Okay, so we're simulating famine, we're simulating hypoxia and temperature extremes. But what about when we actually do eat? Because the concept of adversity applies to our food too, right?
Oh, absolutely.
Because Sinclair notoriously pivoted his diet away from meat. And it wasn't an ethical thing, it was because meat lacks polyphenols.
Right. Polyphenols are basically the organic chemicals that plants make to defend themselves against environmental trauma. And this brings us to one of my favorite concepts, xenohormesis.
Xenohormesis. Okay, xeno meaning foreign, right?
Yep, external hormesis, We are literally consuming the biological stress response of another species to trigger our own.
Which is a crazy paradox when you think about it. Like,
Yeah.
we are constantly told to avoid stress in our own lives, take it easy, relax, but we should actively seek out and eat plants that have been heavily traumatized.
It's a remarkable biological loophole.
Mm-hmm.
Think about traditional matcha tea. Weeks before they harvest it, the farmers cover the tea plants with these heavy shade cloths. They severely deprive them of sunlight.
So the plant thinks it's dying.
Exactly. It senses a threat to its survival, so it overcompensates. It floods its leaves with protective polyphenols to try and maximize whatever tiny bit of light is left.
And then we drink it.
Right. When you drink that matcha, you are ingesting the biochemical manifestation of that plant's panic.
Wow. And Brussels sprouts do the same thing, don't they?
They do. They generate a compound called sulforaphane when their tissues are physically damaged. And grapes produce resveratrol when they get stressed by fungus or dehydration.
So we are just borrowing their chemical defense systems.
Exactly. And if NAD is the gas in the tank that sirtuins need to operate, these plant-derived polyphenols, they act as the accelerator pedal. They hyperactivate the sirtuins so we get the benefits without actually having to endure real starvation.
And Sinclair tries to mimic this synergy with a very specific pulsed supplement stack,
He
right?
does. He uses NMN, which is a direct precursor to boost his NAD levels. He pairs that with resveratrol to press the accelerator pedal.
What about spermidine and glycine?
Yeah, he takes spermidine, which biochemically mimics fasting to induce that deep cleaning autophagy, and glycine to support DNA methylation.
But he pulses them. That's a huge point.
Yes, the pulsing is critical. He cycles them so the body doesn't just get used to it and become complacent again. He does the same thing with the prescription drug metformin or berberine, which is the plant-based alternative.
He takes those to lower blood glucose, right? But he specifically avoids taking them right before workout. Why is that?
Because if the whole point of exercise is to create a spike of acute oxidative stress to trigger hormesis, flooding your system with a metabolic inhibitor right before the workout completely blunts the adaptation.
Oh, I see. It's like putting out the fire before the fire department even gets the call.
Perfect way to put it. Timing the stress is just as important as the stress itself. And honestly, it shows why certain trendy diets fall short for long-term longevity.
Like the keto diet.
Right. Strict keto might give you great short-term clarity and metabolic flexibility, but by drastically cutting out plant intake, you're starving your cellular machinery of those essential polyphenolic triggers. You need those for long-term epigenetic maintenance.
Okay, stepping back for a second. Looking at the trajectory of these AAV2 gene therapies and these intense lifestyle shifts, we are staring down the possibility of pushing average human health spans past 120, maybe even 150 years.
Easily. That's not science fiction anymore.
Right. But if we do that, we aren't just changing biology. We are fundamentally altering human purpose. We're shattering all our sociological structures.
And the immediate reflex from society is usually resistance. People push back on the meaning of life.
Yeah, they argue that radical life extension strips life of its urgency. Like, knowing we have limited time is what gives human existence its meaning.
But honestly, that argument falls apart pretty quickly under scrutiny. Biological youth is the most valuable currency on Earth.
Period. I mean, there isn't a billionaire alive who wouldn't trade their entire fortune to permanently be 25 again.
Exactly. Adding 50 healthy years doesn't make us apathetic. It just gives us the bandwidth to master multiple disciplines. You could have 3 entirely distinct careers. You could truly participate in the lives of your great-great-grandchildren.
Not to mention the demographic cliff we're facing. Living longer solves a huge macroeconomic problem.
Oh, massive. By 2050, the ratio of dependent elderly citizens to working-age people is going to be completely unsustainable. Keeping human capital highly functional for an extra 4 decades might be the only mathematical way to avoid global economic collapse.
Okay, so we're talking about information dictating physical reality. From the epigenetic software in our cells to the macroeconomic data of the species, which beautifully transitions us into the physics discussion from the source material.
Yes, this is where it gets really mind-bending.
Seriously,
Yeah.
the double slit experiment. Let's talk about that.
Okay, so in quantum mechanics, the double slit experiment basically demonstrates that particles can act like classical waves or they can act like solid particles.
Depending on if we look at them, which is insane.
Right. When unobserved, an electron travels as a wave of infinite probabilities. But the precise moment a conscious observer measures it, the wave function collapses. The probability instantly becomes a definite physical particle.
Wait. So just the mere act of conscious observation dictates the physical reality of the matter.
Yes.
So if reality changes just because I look at a microscopic particle, does that mean I am literally rendering the universe in real time just by existing?
That is exactly what Sinclair hypothesizes. He believes there's a greater than 50% chance we are in a simulation. And that consciousness isn't just an accident of evolution, it might be the fundamental architecture of the universe.
The universe organizes matter just to create conscious entities capable of observing it.
Wow.
My mind is totally blown.
It's wild. But bring it back to the biology of aging. If consciousness is the mechanism that collapses probability into reality, and aging threatens to extinguish that consciousness.
Then solving the information theory of aging is way bigger than medicine.
Exactly. As AI starts developing its own novel forms of cognition, preserving human consciousness becomes like a cosmic imperative. We are the mechanism by which the universe renders itself.
Wow. Okay, to synthesize this entire massive paradigm shift for everyone listening, aging is not an inevitable mechanical wearing down.
No, it's an epigenetic identity crisis caused by information loss.
The software gets corrupted, but the original code, the beautiful music, remains totally intact within your cells. And while we are waiting for the clinical trials on viral reprogramming, you hold the power to slow this loss down today.
Right. You can force your cellular maintenance crews to keep the record clean.
By intentionally injecting mild adversity into your life, fasting, panting, eating traumatized plants, you have to actively reject abundance mode.
You have to if you want to protect your cellular identity.
Well, thank you for joining us on this custom-tailored deep dive into Dr. Sinclair's research. It's been an incredible journey.
Yeah, it really has been. Thanks for having me.
But before we sign off, there's one final really provocative thought that builds on that crazy physics concept.
Oh, this bridges everything perfectly.
Think about this. If the double-slit experiment proves that human observation literally alters the physical behavior of particles, and if aging is essentially just the physical decay of microscopic particles inside our cells, could our deep-seated societal belief that aging is inevitable actually be a self-fulfilling prophecy?
That is a fascinating question.
Like, if we collectively observe our own timelines as a slow, unavoidable decay, are we actually forcing the wave function of our epigenome to collapse into senescence? If we truly change how we observe our own biology, could consciousness itself become the ultimate anti-aging tool? Keep pondering that until next time.